Thank you for joining us on our lab tour of the Maltz family Ehlers-Danlos genetics lab. We're going to take you through different components of the lab and show you sort of how we're functioning as a unit. So, come on in. So right off the bat, when you come into the lab, you'll see this sort of cabinet. This is sort of our first line of our genetic study, which is the actual patient samples. In here are saliva samples from members of our registry, of our EDS registry. They come in and they're quarantined for a couple weeks just to ensure no COVID, active COVID vaccine or virus. And so then we'll proceed into the lab. And so there's the great sign, thanks to L&K and Maltz family. Now the only designated Ehlers-Danlos genetics lab in the world. So we're really proud of this. We can't do it without everybody's help, and especially thanks to L&K for the kind donation. So we're going to go this way.
Okay.
So there's a few ways to get into the lab. This is the way that most people come. What we'll see when we turn this way is, number one, we have a lot of artwork on the walls to really demonstrate all the different types of research that we do. A number of these are actually have to do with Ehlers-Danlos syndrome, and it's a representation of what all the students produce in the lab. And so people who come into the lab can immediately see the different areas of research that we focus on. And so back here in the back, we have a cell and tissue culture facility that's embedded within the lab. This is where we take cells from either patients or our animal model with Ehlers-Danlos syndrome and can study it, can study the disease in a dish. And so we have the actual hoods here where we do the work. We have incubators and you'll see that there's cells in here. You can't really see the cells, but you can see the dishes. And these are actually cells from Ehlers-Danlos syndrome animal models that we have growing. And then we can look at the cells and analyze them underneath the scope. That's probably too hard to actually. Can you actually see cells under there with the camera?
Great. You can just use a part of this.
Yeah.
When samples come into the lab, we have to extract the DNA from them after the quarantine period. So over here you'll see all of our saliva kit supplies to send them out and collect samples from patients. All in here. It's really simple. Just a little tube that people spit into, and then we extract the DNA. And we get these little tubes with DNA in them that we use to do all of our sequencing and genetic studies. Christina is helping keep this area very organized. We actually got so many samples that we're adding on more storage to be able to keep track of all the DNAs we've received.
Yeah, so right now we have almost 4,000 people in our registry, getting close to 1,500 people consented and about 1,000 DNAs. So we're continuing to enroll. We have no end dates. We want to just allow as many patients to be involved as possible. So we're going to go to the back of the lab, or back portion of the lab, and this is what we call our innovation station. During the summers, we're fortunate to have an intern program that brings in patients with HEDS. So these are college and post-bac students who have the disease who want to build a career understanding Ehlers-Danlos syndrome. And so this can be future scientists, future physicians, nurses, anybody in the healthcare system. And so we initiated an HEDS intern program. It's now called the Gensimer HEDS intern program after Courtney, who really was the impetus for these studies. And so back here in the corner is an area where the interns, this year we're having 5 interns come during the summer. Next year we'll have 6, and the following year we'll have 7. And so this back area is an area for them to kind of get together, talk about the EDS, what they want to study, how they want to study it, as well as a whiteboard back there that allows them to sort of use as a think tank where they can sort of generate ideas. And so this is the concept of building a patient scientist is really important for us because we need a larger community of healthcare providers that understand the disease, and who better to do it than those who have had EDS. So this summer, in addition to the EDS interns, we'll have a number of other volunteers that range from high school to parents with children with EDS. And so we'll have about 12 to 14 people in the lab with HEDS themselves studying the disease, which is really going to help us accelerate discovery. So in this area, a lot of our molecular biology, biochemistry, protein work happens. We have a lot of different tools in here that allow us to look at various aspects of what proteins are doing, what are enzymes doing, what are the faulty genes in EDS doing, what's the consequence of the mutations. And so Rachel is actually doing what's called a Western analysis, where she can look at specific proteins. And maybe she's doing something with mast cells. Is this mast cells? Yeah. So one of the things that we found in our EDS model, and which is also common in the EDS public, is the mast cell activation syndrome. And so we're in the process of trying to, and that's something we see in our animal model as well. And so we're trying to understand what this gene defect does to result in a mast cell degranulating and having problems. And so one way of doing that is to use this contraption or device and to be able to look at specific proteins. And so that's what she's doing. She's loading the samples directly into these little wells, and
You want a close-up?
they're purple, so you can actually see them.
Yeah, so it sits in there.
Yeah. And they'll run down the gel. There's actually a little tiny gel in there, and the rundown will be able to assay different things and try to understand why the mutations are causing a mast cell.
So this is our histology area where we do all sorts of tissue histology from both animals and humans. We are able to section tissues over here. So this is one of the tissue blocks that someone was using, and cut off little wax pieces that we then mount onto slides to do histology on. So we would come over here and take the tissues that look like this. So these little spots on here are actually pieces of tissue. And we take those and put them onto slides to get something like this. And then from these tissue slides, we can stain for different proteins to look at things like collagen and other components of connective tissue. And here's an example of what some of those can look like. But that's what a lot of the artwork hanging in our lab comes from.
So in this area here is actually part of our transgenic facility, and we use them to actually generate our animal models. So we can take the genetic information that we get from patients, come over here and use a technology called CRISPR-Cas9 to actually put in the human mutation into an animal model, into either a mouse, a living mouse, or into various cells. And that's really the way that the best way that we can validate the mutation as causative. So we can generate an animal model that we can use to not only validate the mutations, but also use to understand the disease and the development and progression of the disease. So really the lab is set up to do everything from collecting the patient samples and the saliva, getting the genetic information, coming here, generating the animal models, and then going around the different aspects of the lab to really provide a holistic understanding, a mechanistic understanding of what's causing the disease. The other thing that's really important about the animal models is now that we have the first model for Ehlers-Danlos syndrome, it not only allows us to understand the mechanism and why does the disease happen, but it also allows a platform for therapeutic development and therapeutic discoveries. We think that that's going to be really important for the future of Ehlers-Danlos research, not only having a diagnostic tool, but also having therapeutic options for these